I became very interested in the idea that, almost like Sherlock Holmes, there has to be an answer to the mystery of why people are ill. And we've had, I think, leaps of innovation that have impacted the pace with which we can integrate biology and chemistry and physics to really start designing drugs almost like engineers. I think for a long time, drug discovery has been very empirical. Think back to people with petri dishes trying to understand basic biology. Now we've been able to industrialize and turn a lot of drug discovery into an engineering problem. Essentially, that is structure-based drug design. It's designing drugs not with a blindfold on, but with supervision now of how proteins are behaving, moving, and interacting with each other. So Schrödinger has been doing fundamental science for about 30 years, trying to, first of all, get the physics right and then code it into software. It's now being used to predict the properties of molecules before you synthesize them. These methods have become so accurate that the ability of the predictions, the computational predictions, to mimic what you're seeing in the petri dish, we can now predict the properties of millions, if not billions, of molecules right at the very start of a project. We can essentially take the blindfolds off in terms of the way in which we're designing drugs. Cryo-EM now allows us to get these images, essentially, of proteins to an incredibly high degree of accuracy and resolution and capture proteins almost in their native form.
So Schrödinger made the decision to invest in cryo-EM because we wanted to broaden our targets in our portfolio to more challenging targets. We need cryo-EM to be able to access that high-quality 3D structural information. So GPCRs, ion channels, large multi-protein complexes. These are target classes that are traditionally really difficult to solve by X-ray crystallography.
Cryo-EM used to be this bespoke technology that was only available to a few labs around the world. Thermo have now democratized this, bringing cryo-EM to thousands of labs across the world. And I think the collaboration with Schrödinger is essentially bringing two essential components of structure-based drug design together so that people can learn not just how to get these highly accurate structures, but also how to apply these predictive methods to those structures to accelerate drug design. And I think one of the shared missions of Schrödinger and Thermo Fisher has been to essentially democratize access to the underlying capabilities that are required to do structure-based drug design. Because while we have discovered amazing drugs that are changing the lives of patients, there are so many patients still left to serve.